For years, the public blamed a single, handsome airline flight attendant for bringing a death sentence to America. Headlines claimed he crossed the country, city after city, knowingly infecting thousands and igniting a global epidemic. They called him Patient Zero. This narrative became the center of a best-selling book and a centerpiece of the evening news. It provided a frightened public with exactly what it wanted: a clean story with a clear villain to point at. But the villain was a clerical error. During early mapping, investigators tagged this individual's case file with the letter O to indicate he lived outside California. On the typewriters used in the early 1980s, a capital letter O and the number 0 were virtually identical. Patient O was misread as Patient Zero. Decades later, when scientists sequenced the virus from his own tissue samples, he didn't sit at the root of the evolutionary tree. He was mathematically in the middle of a pre-existing outbreak. The virus was already mutating and spreading across the country long before he could have been the source. This scapegoat narrative was as destructive as it was false. It allowed the public to focus their fear on a single person, while the real killer, an invisible, complex pathogen, continued moving through the population. The actual investigation began on July 3, 1981, in Los Angeles. Doctors were sounding the alarm about young dying of incredibly rare fungal infections of the lungs, the kind of illnesses usually only seen in people with completely broken immune systems. Dr. Michael Gottlieb was among the first to notice that these patients weren't suffering from a series of coincidental infections. They were all experiencing a total, systematic collapse of their body's ability to defend itself. Researchers in the US and France soon identified a bizarre, newly discovered virus as the culprit. They named it HIV. Standard epidemiology relies on mapping human contacts, but HIV's ability to hide for years rendered these maps incomplete. The investigation moved beyond interviews, looking for the answers written inside the genetic code of the virus itself. To solve this outbreak, researchers treated every viral mutation as forensic evidence, allowing the pathogen's history to speak where patients could not. HIV is an efficient hunter. It doesn't attack the body randomly. It targets CD4 T cells. These are the generals of the immune system, the specific cells responsible for reading threats and coordinating a response. By destroying them, the virus leaves the rest of the immune system without instructions. It searches the bloodstream for a specific docking point on the cell membrane, anchoring itself securely before bypassing the surface barrier to slip inside. Inside, it deploys an enzyme called reverse transcriptase. While most life reads DNA into RNA, HIV reverses the flow, building double-stranded DNA. This viral DNA then hot glues itself directly into the host's genome, sitting silently for years before initiating immune collapse. But this process has a critical flaw. Reverse transcriptase has no spell check. Every time the virus copies itself, it makes random genetic typos, forcing each new generation to be slightly different from the last. Every typo is passed down to all future descendants. This constant accumulation of errors was the exact weakness scientists used to trace the virus back to its origins. This creates a molecular clock. Because typos accumulate at a predictable rate, they act as a biological record. By comparing differences between two modern viral samples, scientists reverse the clock. The more mutations separating them, the further back their shared ancestor must be. To make this clock accurate, however, scientists needed a baseline, a precisely dated genetic fossil to calibrate their math against. They found it in a piece of human lymph node removed in Central Africa in 1960. Preserved in wax in a forgotten archive for decades, it still contained the genetic signature of the virus exactly as it existed 60 years ago. By combining these old samples with modern sequencing, researchers built a chronological map that pointed directly to the era and the location where the pandemic strain was born. The molecular clock revealed that the pandemic strain of HIV-1 jumped to humans roughly around the 1920s, 60 years before the world ever noticed it. This jump likely happened through the butcher of wild chimpanzees for meat. A single slip of a knife, a small cut on a hunter's hand, and a fleeting contact between human and animal blood. That single biological accident was the starting point. The virus found its foothold in Leopoldville, modern-day Kinshasa. In the 1920s, this was a booming colonial city in Central Africa, growing faster than its infrastructure could handle. Social and political upheaval pulled thousands of men into the city for labor, resulting in a severe gender imbalance. This environment reshaped the local sex trade, providing a concentrated network for the virus to move through the population. Colonial powers were building the perfect highway for a bloodborne pathogen. New railways and river steamers allowed people to move between towns at unprecedented speeds, while early medical clinics often reused needles that were poorly cleaned between patients. The virus spread invisibly for decades, its deaths blamed on other causes. By 1960, it traveled from Africa to Haiti, where it established a new, silent foothold. From Haiti, it arrived in New York City around 1970. The virus was already established in America a full 10 years before Dr. Gottlieb saw his first patients in Los Angeles. HIV didn't possess a special ability to conquer the world. It was simply the beneficiary of human colonialism, urbanization, and rapid industrialization, which provided the ideal conditions for a rare spillover to become a global pandemic. This decades-long history of infrastructure-driven spread stands in stark contrast to the myth of patient zero. The biological record shows that no single individual was the cause. The world had been prepared for this spread long before 1980. When we face a terrifying, faceless threat, we naturally look for a scapegoat. We seek a villain to blame, because the truth that we are part of a complex, often chaotic natural world is much harder to process. Pandemics are not acts of human malice. They are the result of biological proximity and the mathematical probability of a virus finding exactly the right tools to survive in a new host. As we continue to push deeper into wild habitats and move around the globe faster than we ever have, more spillovers are certain. Our best defense is to look past comforting myths and focus on the cold, scientific truth of how these viruses actually move.